Hello, everyone. I'm Andy Berglund, Director of the RNA Institute and Professor of Biological Sciences here at the University of Albany. And it's my pleasure to serve as the moderator for today's RNA Collaborative seminar series. I will introduce our first speaker, Shenglong Zhang from the University of Albany. He's an Associate Professor in the Department of Chemistry and a member of the RNA Institute. Before we get started, I just want to remind people to put your questions in the chat. At the end of Long's presentation, we'll have a short period where we can do live Q&A. Take it away, Long. Thank you.
Thank you, Andy, for the introduction. Can you guys see my slides all right?
Yep, you're all set.
Great. It's great honor to give this seminar. As Andy mentioned, I'm a faculty member in chemistry and RNA Institute at the SUNY Albany. Today, I'm going to talk about the next generation mass spectrometry-based sequencing. This is a new platform we have been developing for exhaustive sequencing of RNA and unbiased sequencing of RNA modifications. We also expect that this can be used for global profiling of RNA and the modifications in human diseases. If we look back, like in the year 2003, and this was a very important year in our field. And this was the year that the Human Genome Project was completed. It took 13 years and billions of dollars to draft the first sequence of our human genome. Now, a comparable project at a similar scale would be the Human RNA project to draft the first sequence of all human RNA and their modifications. We can call it the second Human Genome Project for RNA, or like Human Epitranscriptome Project. However, this project has not yet exploded, mainly because we don't have a technology compared to Sanger sequencing that was used in first Human Genome Project. In fact, we actually don't know the true sequence of RNA or RNA known. And that's the identity and the location of each RNA nucleotide building block modified a lot in the RNA molecule. You may wonder, what's the problem? And this is more like when we play jigsaw puzzle. If we have very limited puzzle piece, it will be difficult for us to see the whole picture. And this is the problem with the current RNA sequencing technology. Current technology can sequence only a subset of RNA sequences or modification, not their entirety. As such, we don't know the true sequence of RNA, and we can't completely understand the RNA sequence and function diversity. We know, like Illumina-based or next generation-based RNA sequencing, they are very powerful. They can read the full nucleobase ACGU in high throughput fashion. However, these methods are indirect. They can't faithfully reflect the initial RNA counts or sequences in the sample. You may say a NaNoPo is a direct method, but the NaNoPo depends on ligating RNA to the sequencing at depths. We know the ligation, the yield could be very widely. So for the RNA that can't be ligated to the sequence at depths, and they can't get the sequence. So as such, we actually don't know how many unique RNA molecules or sequences in a sample. If we look at the RNA modification, Typically, one method can only map one specific RNA modification type. Collectively, now we can map around 12 RNA modifications are listed here. However, this is just like tip of iceberg. We know now they have more than 170 different RNA modifications has been discovered, and this number is still increasing. Now some are showing on the right. For those individual methods, there's no path that they can be used to sequence all those RNA modifications. This actually is one of the most significant challenges in our field. And recently, this challenge has gained attention from both NIH and National Academy of Science. The field is calling for solutions for direct sequence of all modifications, for capture RNA sequence diversity, and for more efforts that could allow us comprehensively sequence like RNA epitranscriptome. We know it's not trivial to sequence 4 nucleotides G, C, GU in an RNA molecule. How can we sequence all different types of modification together with this 4 nucleotides? And this is why we propose mass spectrometry-based sequencing. Because for sequencing, typically we need to have a signature tag to identify each nucleotide. In terms of a tag, nothing is better than mass. Mass is a natural tag, both for clonic and modified nucleotides. This includes unknown nucleotide modifications. It would be difficult for another method now to identify all those RNA modifications. You may be wondering, how can mass be used to sequence all those RNA modifications? In fact, conventional mass now even have difficulty to sequence RNA. We know that tandem mass spectrometry, also known as MS, has been used to characterize RNA and RNA modifications for decades. Coupled with the liquid chromatography, they are considered as the golden standard for RNA modification analysis. However, to use those methods, typically we have to purify the RNA, digest them to a single nucleotide or single nucleosides. They can't provide location information, or they need another methods to provide this sequence information. So that's why we are developing next generation mass spectrometry-based sequencing. We would like to empower mass so that they can be used for de novo sequencing of RNA. We want to develop this toward a high throughput. We also want this to be used for quantitative mapping of RNA modification that are modified partially, more than 100% at the last sites. And how can we de novo mass sequence of RNA? We know single sequencing has de novo sequencing capacity. That's because of a single sequencing relies on the ladder generated by single dideoxy reaction. This, of course, is the Nobel Prize work. If we can generate a similar ladder for RNA, and then we can use mass spectrometry to de novo sequence RNA, the base calling can be made by comparing the mass differences between two adjacent ladder fragments and then comparing them to the known nucleobase to read each nucleotide in RNA sequence. One way to generate a ladder like this is to cut RNA once but randomly among RNA. This can be achieved by controlled hydrolysis. And this will generate two ladder pool, 5' ladder pool and 3' ladder pool. The sequencing can be read from each pool and they can reaffirm each other for sequencing. And this is typically how our data looks like. If we plug mass against retention time, we can see this two-linear curve. The small ladder fragments come out early with small retention time. Big fragments come out later with big retention time. So this resembles this sigmoidal curve. And also we know the 5' ladder and the 3' ladder. The 3' ladder doesn't have a phosphate the 3' ends. So the 5' ladder on the top of this two ladder adjacent ladder curves. So this will allow us to de novo sequence of RNA. We can read from 3' to 5' direction or 5' to 3' direction. The advantage of this approach is that we can not only read ACG4 nucleotides, we can also read sequence those RNA modifications. However, this method also has limitations. Mainly, it can be used for only synthetic RNA. They can't handle complicated cellular RNA samples. The reason for this is like, for one RNA, you have this two letter adjacent to each other. So if you have many RNA samples, RNA molecules in the sample, and they're going to have lots of overlaps. So how to really push this method to allow mixed RNA sample be sequenced, or to increase the sequencing throughput. Now we have many different approaches after 10 years hard working. Of course, if we have a higher, better instrument, if we introduce automation, that could help to increase the throughput. But through years of hard work, we developed a lot of strategy to help increase the throughput. But for the interest of time, I'm mainly going to talk about the MassSum. MassSum is a computational data separation strategy. Remember, we rely on the formic acid hydrolysis to generate a ladder. If we look at the any pair of this ladder fragments generated by this one-cut event, then MassSum is a constant. This is equals to the mass of the RNA plus a water molecule. If we look at this, the 5' ladder and the 3' ladder, if we look at the any pair generated by this one-cut event, again, the MassSum is a constant. Because of each RNA has a unique mass, so we can computationally separate all the paired ladder fragments for sequencing each RNA in a mixed sample. So we first test this on tRNA. tRNA is a difficult one to get sequenced by high throughput sequencing technology because they have many RNA modifications. tRNA is also difficult for traditional mass to sequence them because traditionally mass, they requires a single tRNA sequence, which is very difficult to get. And this is the showing here, this is the yeast phenol tRNA we get from sigma address. Supposedly 76 more, and with CCA is the major, should be the major product. But clearly, the major product, product is not this. Is the one 75 more with the A missing at 3' ends. So despite the difficulty of physically separating this tRNA with the MassSum, we can computationally separate them, the ladder fragments using their unique mass. So with MassSum strategy, this can help to reduce the need for physical purification, allow mass to sequence any RNA in a mixture directly. And this could also help increase the throughput of mass sequencing, and now sequencing potentially unlimited number allowed by the instrument. That also paves the way for large-scale mass sequencing biological samples. One of the challenges in mass sequencing is like how to differentiate the pseudouridine from uridine. We know they have identical mass. Mass alone would be difficult to differentiate them. So we treat the RNA with CMC, and this is going to selectively react with pseudouridine, not uridine. And they're going to form this complex. The mass is going to increase to 252. After the acid degradation of this RNA, in their ladder fragments is going to move to the bigger mass region because the mass increase. And also this complex is more hydrophobic. So after the acid hydrolysis, their ladder fragments is going to shift to the big retention time region. Because of the yield typically is over 100%. So we're going to see two curves. One is corresponding to converted one is corresponding to a non-converted one. This is what started from pseudouridine. And this is our data. So you can see with this, we can easily identify what the pseudouridine is located. If there are more than one sugar uridine, we can also nucleate this easily. Interestingly, the branch in this 2D mass against retention time curve suggests a partial single nucleotide substitution. And the curve in the branch indicates coexistence of two nucleotides, signifying partial RNA modification. This actually is very significant because this can help us, the branch could help us to identify partial RNA modifications, which right now is very difficult to identify in the first place. So this is a huge advantage of our technology. So once we have our RNA sample, we divided this into two portions. One is go directly to the LC, and the second half will go through this acid hydrolysis to generate a ladder. So if we have a partial modification, say a partial methylation, and the intact level, we should be able to see two mass, their differences should be 14, right? After the acid degradation, we can find a ladder, and we can pinpoint, like find the branch that will identify, help us to pinpoint why exactly this modification is located. If there's a lot of AG transition, and we should see a lot of two masses. The mass differences should be 16. Right? And after the acid degradation, we should see four branches that corresponding to this two, one methylation, one AG transition. And this is the one, the branches when we sequence in tRNA glue from mouse leaf sample. As you can see, we can there's mass differences indicated there's A and methylated A coexist in this position, position 58. So we can know their identity and the location. We can also further determine the stoichiometry because the ladder fragments we use for sequencing also carries relative abundance information. So we can export, like extract the ion chromatography shown here, and integrate them to calculate their stoichiometry. And I have to say, and this is not the absolute quantification. This is just a relative stoichiometry of modified one versus unmodified one because they are coexist at the same condition. So therefore, they can work as the internal reference for each other, allow us to calculate the stoichiometry. And with this standard curve also verify that this is an accurate method. So with all this development, we can now use mass to sequence full-length tRNA. As you can see, we cannot just read the ACGU full nucleobase. We can also sequence all the modifications simultaneously. Among these 10 modification sites, we find 3 of them are not modified 100%. We further determined their stoichiometry are shown here. To our knowledge, and this was the first time we can directly sequence of full-length tRNA with this level of details for RNA modifications. And this will, of course, also help us to review the true sequence of RNA. The RNA, tRNA specifically, they have lots of methylation, like isomeric methylation. If the methylation is in 2O position, like shown here, and we can differentiate this, because if there's a methyl group at the 2O position, they're going to block our acid hydrolysis chemistry. Therefore, we should see a gap. So that will help us to identify this 2O methylation. But there are also a lot of type of methylations as well. And this, of course, is very challenging. So today I'm going to talk about our recent efforts to use RKB enzyme, the specificity of RKB enzyme and those lowering methylation to help us to differentiate those isomeric methylations. So before RKB treatment, we can see 58 position, there's only one curve, there's methyl A there. But after RKB treatment, and this curve almost completely disappeared. And they were converted to regular A at the 58th position. So we will know at the 58th position, their probably is M1A and not M6A. Similarly, we can also differentiate at the position 9. This is M1G and not another methylated G. And this is also allow us to track the stoichiometry change before and after the RKB treatment. And this capacity actually is very significant. That could address big challenge right now in the field, potentially allow to monitor RNA modification dynamics and map RNA modifications that have altered the stoichiometry in different cellular or diseases conditions. The sample I was talking about are mainly highly purified tRNA. Our method can also handle complicated sample like total tRNA. Because each tRNA has a mass around like 24,000 Dalton. So you can see in this picture, each dots here indicated as it's one tRNA. We can calculate their theoretical mass using those data mass, and then the ones that can match with the mass calculated from database are shown in orange. But of course, there's a lot of ones that can't find a match. That may indicate there's a new modification or even new sequence. So we really need to sequence them to verify that. So after the sequencing, you can see the data is very complicated. Ideally, we want to de novo sequence them all, but our algorithm is still developing. We can't sequence full-length tRNA in each tRNA in the sample, but we can read out the partial sequence of each tRNA. So with this information, and then we can search our database and to find out what sequence this sequence belongs to, what tRNA this sequence belongs to. So with this input, and then we can then find more letter fragments in our data. Say this is a tRNA as acidic acid. The letter indicates the nucleotides has been verified by our letter fragments. The letter in box indicate the modifications that have been verified by our letter fragments. So one by one, and we can read out, we can conform those RNA nucleotide and the modifications. Of course, not all of them are modified 100%. We further determine that as stoichiometry. So with our approach, we can determine the identity location and the stoichiometry of each tRNA nucleotide or modification in this total tRNA sample. However, they also have, like I mentioned, they also have the tRNA that can't match with our current database. And here is one example shown here. We find a set of mass that can't match with the tRNA reported in a database. And then, of course, we would like to read out the full sequence, and that will be need a new technology. We have been recently developed exhaustive sequencing. Unfortunately, I don't have time to talk about this method, how it works, but I can briefly talk about the result. This is very powerful tools that could allow us find those tRNA that haven't reported in our database. So pretty much we identify the new tRNA. So this is the tRNA gene, yeast tRNA gene we recently find. So again, with our technology, we can read those short segments. For example, this is segments we first read out. And then we cross-verify with the sequence predicted by the yeast genome. And with the information, and then we can get more letter fragments. You can see here was almost covered entire tRNA. And we can further determine their not just ACGU, but what are some modifications as well as their stoichiometry. And we also verified like the wobble position. They have a modification shown here. So this is one example. The other example is shown in this slide. As we know, there are many like unknown modifications in database. For example, for this tRNA prone at this wobble position, we know this is a modified U, but we don't know what this is. But with our approach, we can find this is a 5 carbomoid methyl U at this wobble position, and that is very clear, can help us identify what this modification it is. And we also find at position 27 in database it says this is the regular C, but our data actually shows this is a more methylated C and methylated 100% at this position. So our technology can help address this non-standing puzzle in RNA field, and also lead to new discovery that haven't reported in those database. So we're very excited about this. We're also trying to push this method to sequence a lot of RNA types beyond tRNA. So this is one of the efforts we made recently. Is we can use gel to purify the small ribosomal RNA and call them out and then use mass to sequence them. We're still actively working on this. Hopefully next time I can talk more about this. We are also working with the industry. We funded a company in direct-seq biosciences. We would like to get this technology commercialized. In my point of view, there are 3 major RNA sequencing platform technology. Of course, we know next generation based RNA sequencing led by Illumina, or nanopore based sequencing led by Oxford and Nanopore. Those are very powerful tools. They can read ACGU follicular base in high throughput fashion, but they can only provide limited or no RNA modification information. And this is the area that our technology are good at. So it's really providing a complementary approach for those high throughput methods. All this is just the beginning. We have so many things we can do. I don't know how much time that I have. I probably need to speed up. This is my last slide. If we look at our workflow, there are 3 major parts. So we have our RNA sequencing chemistry. We also need to generate a ladder, and we also need to collect the mass spectrometry data for ladder, and then finally we need to find those data and read out those data. There's so many things now we need to optimize. For example, the mass spectrometry is not mainly used for this purpose, but we can. There's so much work room for us to optimize for this purpose, and also we're actively working on to get better algorithm tools for data analysis. So our goal is really trying to reach sequencing capacity the cellular sequencing that has. Cellular sequencing is the one that is not a high throughput sequencing, but this is the one that is used for to complete the first human genome project. This can be a standard known approach, and this also can be used to together with high throughput sequencing technology. It potentially could allow global profiling RNA modification, even there are changes in human diseases. And like I said, this can help to reveal the true sequence of RNA, quantitative mapping RNA modifications, even at tissue or cell-specific level. And this can be an organic approach complementary to high throughput methods. So with this, I would like to thank my team. Used to be in NYIT, New York Tech, and now it's in SUNY. I also would like to thank my previous advisor, especially my collaborators who provide precious sample for our study. I also want to thank NIH for the funding support. Finally, I want to thank your attention. I will be happy to answer if there's any question.
That was spectacular, Long. We have a couple of questions, but we are tight on time. So I'm going to summarize 2 of the 3 questions and allow you to answer, but then we do need to transition to Sweta. And Long, you can answer the questions in the Q&A box. But briefly, 2 individuals, Isabel and Navia, asked, what's the longest sequence that can be basically read using the MassSpec? So the length, I guess, is the key question. I was actually quite curious too. Like, is an mRNA going to be possible? You were talking about ribosomal RNA.
Yeah, that's a good question, Liz. So our current read length is about we can comfortably sequence RNA like 100 nucleobase now. So for longer RNA, like ribosomal RNA, message RNA, we are also currently working on. So we have to fragment them into the fragments in their range. I think this is also a doable, popular way to handle longer RNA.
Well, let's give Long a round of applause. I know virtually, but I'll actually clap. Thank you, Long. That was spectacular. You have a lot of questions in the Q&A. So Long, please respond to them while Sweta gives her presentation. And again, great stuff. Okay, so it's my pleasure to introduce Dr. Sweta Vangaveti. She's a principal scientist and a research faculty in the RNA Institute, and she's going to tell you about her research. Take it away, Sweta.
All right. Can you see my screen?
Yes. Although it's not in...
Yeah.
Yeah, put it in...
Working on it.
Okay. Yes, now you're all set. Thank you.
Okay. Good morning, everyone. Thank you for tuning in. I'm Sweta Vangaveti. I'm a computational biologist and a research scientist here at the RNA Institute. And sticking to the theme of the session, I'm also going to talk about RNA modifications, but we have a different approach to studying them. We are looking more at the structure-function relationships of RNA modifications using tRNA phenylalanine as a model system, and we use molecular dynamics simulations to do that. So before I get started with the actual talk, I also wanted to give you a brief overview of all the other things that my lab does besides looking into modifications. So a large part of our effort is focused on developing small molecule therapeutics. So studying interactions between small molecules and nucleic acid targets, including both RNA and tRNA. So one of the projects that we have is studying the mechanism of action of small molecules in myotonic dystrophy. If you don't already know, the RNA Institute is part of the New York State DM Center. And so we focus a lot on myotonic dystrophy. And I'll probably not have time to talk about the other stuff, but I just want to give you an overview. And then another small molecule project that we have is targeting T boxes in bacteria and using that as a novel target for antibiotics. We also have a lot of projects involving DNA nanostructures with Dr. Arun Chandrasekharan at the RNA Institute. Most of these projects are collaborative, so we have a great environment here that really helps. We also have, we are also studying protein interactions. So one of the examples is looking into molecular mechanisms of Alzheimer's, where we have A-beta peptides and we dog them, we study their interactions against neuronal receptors. So today, I'm going to talk about, last but not the least, the RNA modifications. And we not only study tRNA, but we also look into other modifications in other forms of RNA. But tRNA seems like a great model system, as Dr. Long already mentioned. So a lot of the work that I'm going to show you today, the simulations were actually performed by Rose and Rebecca. who are both graduate students in the Department of Chemistry. Rebecca has since graduated, but Rose is graduating soon, and she's very talented. So if you're looking for a postdoc, please reach out. All right. So here's my analogy for RNA modifications. If you think of RNA as your smartphone, RNA modifications can be different accessories that you use on your smartphone to protect it, to enhance its features. So I just thought of a few examples which might help you, where let's say you have 5' and 3' caps of the RNA that protect it from degradation. That could be like your screen protector that is protecting your screen from scratches and damage. And you could also think of other modifications that impart conformational stability. Pseudouridine comes to mind, which has been established as a nucleotide that helps maintain the structure of the RNA. And then inosine might be like a new app that you put on your iPhone, that you put on your phone, which imparts it new features, just like inosine would expands the base pairing capability. So here are just a few examples, but there are obviously tons of other modifications, and we really don't have a clear idea of what they do. There are some studies in ribosomal RNA, in translation, in mRNA that we know of, where the structure and function relationship is established in those contexts. But there are several others which sometimes we know they are there, but we don't know what they do. And so simulating RNA modifications is a great way to study these interactions, just because it's cost effective. We all know how expensive RNA is and how expensive modified RNA can be. Also, when you're conducting thought experiments, let's say you have an RNA that lacks a modification, doesn't bind to a target, then you cannot really solve its structure, but you could potentially use molecular modeling to see how these modifications are affecting the structure or the interaction that you're trying to study. Also, if you want to introduce non-native modifications, where let's say you don't have the reader-writer enzymes, or you don't know if you can exactly insert them in a certain position, but you want to test them out, molecular modeling is a great way to do that. Obviously, if all this is so good, why aren't we all doing MD simulations? Because obviously there are challenges. So I kind of think of it as a chicken and egg problem. So obviously, the experimentalists rely on simulations and modeling to provide insights into the structure and the interactions. But as simulation people, we rely on experiments to get those structures, or at least validate what we see. So I could run a simulation of an RNA today, but whether it is doing what it's supposed to do, or if it will act the same way it does in the simulation as it would in the experiment, to determine that, I rely heavily on my experimental collaborators. So obviously we have made progress in both simulations and experiments, but we are still not at the point where we can convincingly use simulations to predict things. But when simulations are able to validate or at least replicate experimental observations, that gives us confidence that we are moving in this right direction. So to study modifications, we chose tRNA as our model system, as most people in the field do, just because, one, they're heavily modified, and there are structures available of the tRNA with the modifications, which makes it a great tool for MD simulations. So on the left here, for those of you new to the tRNA structure world, the cloverleaf structure should seem familiar to most of you. So you have the 4 stems that are joined by this junction here. There's the acceptor arm. This is where the amino acid gets attached. There's a T-side C loop. Now the tRNAs have so many conserved modifications that one of the stem loops is actually named after the modification, which is the pseudouridine. So that's interesting. And then you have the anticodon loop, which is the business end of the tRNA. This interacts with the mRNA. And then there's a D loop, which is also because you have 2 dihydrouridines on this loop here. So this is how the 2D structure looks like. But if you look at the 3D structure, this is the classic elbow structure of the tRNA. The T loop and the D side C loop come together, And this interaction basically helps it fold into the structure that we all see. Now, there have been several groups that have worked on simulating different modifications, but most of them work with modifications one at a time. We ourselves have also done some. So Stacy Wetmore's group has looked at modifications on the 37th position. We have done some simulations where we looked at modifications on the 34th position and the 32nd position. So I'm giving you, I'll give you two examples here. So this is tRNA lysine anticodon stem loop. On this column here, you have the two codons, AAA and AAG. Those are the two codons that code for lysine. Now, if you have an anticodon stem loop that has this sequence with the unmodified U34, it can decode both AAA and AAG. But as soon as you introduce a granulation, which is this sulfur group with the 12 carbon chain with two double bonds in here, it suddenly does not like the AAA so much, the cognate codon, but it binds to the near cognate codon. So obviously you can tell from here that it's the modification, it loses one of its base pairing atoms. And we performed simulations where we introduced this modification into the mRNA tRNA context within the ribosome. And we showed via these simulations that all the interactions, so the geranyl group, even though it's long, it can be accommodated in the A site of the ribosome. And it also maintains all its native interactions that it has with the ribosome. I'm sorry, I didn't mention this is the bacterial ribosome. And also we showed that if you replace the U34 here with the geranyl group, then the number of the interactions that it has, the strength of the interaction between the 34 and the third position on the mRNA, it's stronger. Obviously for the AU, when it's unmodified, it's strong. But as soon as you introduce the granulation, there is no interaction that we observe in the simulation, versus the GU interaction is as strong as the AU interaction. So this is one instance where we were able to explain the experimental observation, but also show how the geranyl group gets accommodated in the site, even though it seems very big. And here's another example. This is a collaboration with Dr. Agras. So this is where we had, this is for tRNA arginine. So the stem loop has the inosine at the 34th position. And when you have inosine, it can base pair with all three nucleotides A, C's, and U's. And so you see ribosome binding. But when you introduce a thiocitidine on the 32nd position, you suddenly see that there is no binding with the CGI. And this was kind of perplexing because there was no modification in the anticodon region, which was directly affecting this interaction. So we used, first of all, we were lucky that there was a structure of the inosine with the U and the A. So we could look at those structures, and we figured out that the U-turn is different. So this is the canonical U-turn, where there is a conserved U33 in the anticodon stem loop that interacts with the opposite end of the anticodon stem loop, or G36. But this is when you have a purine and a pyrimidine interacting with each other. When you have two purines, which would be the case when you have inosine and A interacting with each other, this U-turn is slightly shifted. Now, what this means for the rest of the loop region is that it gets a little deformed. And so, but the sulfur, the oxygen that is present in C in here, that when you change it to a sulfur, then it no longer can establish this cross loop interaction. And so we showed that by simulations that it destabilizes the overall stem loop. So these were two examples where we had we studied single modifications. So now we got a little ambitious, and we wanted to see how well we would do with our parameters if we looked at modifications as a whole and how that affects the structure of the tRNA. So this is we simulated three different systems. So one of them here is the native structure of the tRNA. So the modifications are shown in deep orange here. And then we had an unmodified system where we stripped the tRNA of all its modifications. And then we also had the unmodified system with magnesium ions. And the reason we put in magnesium ions is because we wanted to see if it is able to replicate some of the effects that the modifications have on the structure. So briefly, our computational workflow, we start with parameterizing these modifications. And the reason we do that is you can find some parameters for the modifications in the online, but most of them aren't available or they haven't been validated. So, and by parameters, I mean, what charges do the atoms have? How strongly are they interacting with each other? What are the bonds like? What are the angles like? And so on. So we do most of the parameterization for the modified nucleotides in-house. And then for structure generation, in this case, this is a tRNA phenylalanine. We already had the modified structure, and then we generated the unmodified structure by just removing the modifications and replacing them with their canonical nucleotides. So for example, a pseudouridine would be replaced by a uridine, and M1A replaced by an A, and so on. And then we performed molecular dynamic simulations. So we did the single temperature simulations just to get a feel for what these modifications are doing. But we also wanted to see to enhance the conformational space that we are sampling. We used replica exchange molecular dynamics, which is essentially running the same system at multiple temperatures. And after a certain point of time, you perform these exchanges between, let's say, a lower temperature and a higher temperature system based on certain criteria. If they meet the criteria, the exchange happens. If they don't, the exchange doesn't happen. What this allows is for a lower temperature simulation to access structures from conformations from a higher temperature simulation, which would not otherwise be possible just because it would have to overcome large energy barriers. So using replica exchange molecular dynamics just gives us, long story short, more sampling of the conformational space. All right. So once we had our simulations, the first thing we did was looked at these two parameters. So I'll walk you through this plot. On the y-axis is the root mean square deviation, which just is a number that shows how far away the original structure, the simulated structure is from the original structure. So for all of those simulations and each of those snapshots, we calculated the root mean square deviation, or RMSD. And then on the x-axis is the radius of gyration, which essentially measures the compactness of the molecule. So if you have something that was really close to the elbow structure and also was very compact, you would see it here. And if you had a structure which was really far away from the original structure or was very open, you would see it here. And so just to give you an example, this is how the tRNA structure looks like in this little white blob here. Also, the colors here, the darker the color, the fewer the number of structures we observed for that particular conformation, and the brighter the colors. In this case, a white indicates that we had a large proportion of our trajectories had the structures that had these RMSDs and these radius of gyrations. So in case of the native structure, we found a native simulation. We found that the tRNA structure had very distinct transition states. The secondary structure of the 4 loops did not open much, only the central junction open. And we could track the unfolding to certain conformation states. So you have like one white blob here, one here, and then another one here. However, when we move to the unmodified structure, this is something that has no modifications, we found that the distribution was more diffused. So there were several conformations that the tRNA was trying to sample as it was moving from lower to higher temperatures, so as it was unfolding. And these structures also looked less defined. They had fewer, the stem loop, the anticodon stem loop, opened up early on in the simulations and stayed that way throughout as we were moving towards the more unfolded structures. And when we looked at magnesium, even at 10 millimolar magnesium, we saw that the structure was rock solid. Now, this could also have to do with like how strong the magnesiums are interacting, and it might be a force field issue that we might have to look at, but it looks like the magnesiums are at least stabilizing the tRNA structure in ways that the unmodified ones cannot. So here's the trajectory of the two simulations. On the left is the native with 10 millimolar magnesium, and on the right is the unmodified with the 10 millimolar magnesium. And so you can see that on the left, the structure stays almost stable throughout the simulation. On the right, the unmodified one starts to open up and fold. But if you have magnesium in there, it still maintains its clover leaf structure. So it is able to prevent it from completely unraveling. So next, what we did was we calculated simulated annealing, simulated melting curves, and this would be similar to what you would see in a UV experiment. To do that, we had to categorize each of our trajectories into each of our snapshots into folded or unfolded conformations. And to do that, we used conserved Chi-C interaction between the T psi C loop and the D loop at the elbow here. So if the distance was below 15 angstroms, we used that as a cutoff. If the distance was below 15 angstroms, the snapshot was considered folded. And if it was above 15, it was considered unfolded. So for each temperature simulation that we ran, we found the fraction of folded conformations throughout the trajectory. And then when we plotted this, we saw that for the native, which is this one, the orange one here, the native conformation with the modifications has a melting temperature of around 387 Kelvin in our simulations. The unmodified one was at 365. And then once we added high concentrations of magnesium, it went upwards of 425. So what this is telling you is that removing the modifications from the tRNA reduces its stability, but it can be overcome if you have enough magnesium in the system. So we were curious to look at what interactions are basically being affected. Of the 14 modifications that the tRNA phenylalanine has, 5 of them occur at the D and the T psi C loop. So that's where we focused on. So this is all the interactions of the D loop with the T psi C loop at that elbow junction. And this is in the crystal structure. We were, for our simulations, we were able to show all of those interactions were maintained. This validates our simulations. At least it shows that all our the parameters that we use for our modified nucleotides seem to replicate or reproduce what is observed experimentally. But as soon as you remove these modifications, here's what happens. So these two Ds are now replaced by Us. And then this G, which was able to establish a cross-loop interaction with the T psi C loop, can no longer do that because the U's can stack on top of each other. So the difference between dihydrouridines and uridines is that uridines have a planar surface versus dihydrouridines have an unsaturated ring. So we think that the stacking interaction prevents the interaction between the D loop and the T psi C loop in this context, which is something interesting. We also looked at the magnesium ion distribution around the tRNA and looked at it as a function of magnesium ion concentration. And so on the left, with the native, you can see some of the magnesium ions bind in regions that have modifications. As you increase the concentration of the magnesiums, they are just everywhere and they start to bind more non-specifically. But this is where you get the rock solid conformation. So what we think is magnesium ions are can stabilize the structure of the RNA, tRNA, but they do that non-specifically compared to modifications that would stabilize the specific regions of the tRNA. Here's another example where we looked at the anticodon stem loop. On the left, the modified nucleotides, with the modified nucleotides, the magnesium ion that was also present in the experimental structure, the crystal structure, remained bound throughout the simulation. But in the unmodified system, we saw a loss of that magnesium binding, ion binding site. So what we think is that the modifications actually facilitate the binding of magnesium to certain sites, which is lost as soon as you get rid of the modifications. So in summary, I wanted to tell you that so the first thing that we found is that lack of modified nucleotides in key regions of the tRNA disrupts that hydrogen bonding pattern between the T-Si-C-D loop, which is necessary for maintaining the tertiary structure of the tRNA. That RNA modifications play an important role in creating precise binding sites for magnesium ions to bind and stabilize the structure of the tRNA, which goes missing when you remove those modifications. And even though high concentrations of magnesium can potentially stabilize the tertiary structure of tRNA, even in the absence of modifications, the binding tends to be mostly non-specific. So where are we going with this? So right now we have at least we know that our parameters are great and that we are able to reproduce the experimentally derived structure for the modified tRNA and also explain some of the effects of the magnesium and the modifications on the structure of the tRNA. So now what we want to do is try to model the same system with either different lengths to see how each part is affecting the structure of the tRNA, and also strip it of one or two modifications or introduce only one or two modifications and see how that makes the system evolve. And so with that, I would like to thank my collaborators and lab members and funding sources, and I'm happy to take any questions.
Spectacular talk, Sweta. Thank you. We do have time for a few live questions, actually, if someone wants to ask a live question, or you can put them in the Q&A box. Trying to see if I see any hands up. Liz or Lindsay, if I'm missing something, let me know, please.
I don't see any hands up right now,
Okay.
but we do have a couple of Q&A.
Oh, there they are. Sorry, I missed it. I was in the wrong thing. I was in the answered. Okay. Oh, no name, anonymous. How are the magnesium ions being described in the simulations?
Yeah, so so we use Nadine Schwartz parameters, and those parameters include the at least replicate the behavior of the hydration shell around magnesium. So they are closer to what real magnesium ions would behave like compared to just modeling them as divalent spheres. So I hope that answers your question.
That's great. And I think it's related to a question from Gosheng Ku. At physiological magnesium concentration usually lower than 10 millimolar, what do you expect the result when lower concentrations is used in your simulations? But you basically just answered that. Is that true?
Kind of, yeah. So we did not look at any lower magnesium ion concentrations because even at 10 millimolar, they seem to overstabilize the structure of the tRNA. But I would expect at lower magnesium ion concentrations, we would see just the specific binding sites if we were looking at the native construct. And if we were looking at the unmodified construct, it would be less stable. So essentially, I'm just speculating here because we haven't done the simulation yet. But I would think at lower magnesium ion concentrations, the unmodified structure would be much less stable, suggesting that unmodified tRNAs are harder to find in cells.
Okay. Which force fields were used?
Yeah, sorry, I should have mentioned that. So for the RNA, we use the Chen-Garcia force field, which is developed by Alan Chen here at RPI, and then he's at the RNA Institute and works closely with me. And then for magnesium ions, we use the Nadine Schwartz force fields. And then for water, we use the TIP4P evolved water.
Great. And I think the last question, have you tried using IMS to experimentally verify?
So the reason this project started was because we had Rebecca, who's the student here. She had some results from IMS-MS, and that's why the whole looking at the radius of gyration, because you can tell how expanded or how compact the structure is. But unfortunately, she graduated before we could get all the data from the IMS-MS. But yeah, if there is someone who's interested in doing this, we would you'd be thrilled, because there are combinations that we want to try where we remove certain key modifications that we have identified and see how that affects the overall structure. So yeah, thank you for that question.
Well, it looks like we've timed this perfectly. It just clicked over to 11 o'clock. Long and Sweta, thank you for wonderful talks. I encourage everyone to come to the next RNA Collaborative event on December 4th, and I don't remember the exact time of when that is, but I'm sure you can look it up. Thank you again, everyone, for joining us. Great to see everyone virtually. Bye-bye.